So what we've been talking about with muscle so far is, right, we went back, we looked at how a skeletal muscle contracts, gets shorter, pulls two things together, right? And in the case we've been talking about, the arrangement of those skeletal muscles were attached to, say, two bones, right? Or some facial muscles in the skin, right? And so when they contract, they pull those two things together, right? That's skeletal muscle. And skeletal muscle, where's its location? Its location is on the skeleton, right? That's the whole purpose of it. Whereas your cardiac muscle, so that's all over the body. Cardiac muscle is only in your heart, right? It's just that little lump of tissue right in the middle of your thoracic cavity right there, right? So that's where the location, right, making up most of the meat of your heart, right? Cardiac muscle tissue. And then the other type of tissue, which we're going to run into quite frequently for the rest of the semester, is this smooth muscle. And we're going to run into it because it is going to line, say, your bronchioli tubes of your respiratory tract. It's going to line the blood vessels in your arteries and veins. It's going to line your GI tract tube, right? So there's going to be this layer of smooth muscle that's going to be able to kind of constrict, cause this squeezing force, and like move things along the tract or constrict things, right? So smooth muscle, that'll be a big sort of, we'll run into it over and over again during the rest of the semester. Those are two, differences where you're going to find those types of muscle, right? But they all do a similar thing. They're contracting in some way and creating a pulling or squeezing force. So the control over that, whether a muscle contracts or not, is different, depending on what type of muscle. For all the stuff we've been talking about, we went over the neuromuscular junction, where these spinal motor neurons in the spinal cord were sending out a signal telling this muscle to contract, right? And so when we want to flex your biceps, you can do that voluntarily. When you want to smile, you can do it voluntarily. Even though you could do a lot of these unconsciously and all that, you have voluntary control over when those muscles contract for the most part, right? So the regulation, the control of this muscle is voluntary. Cardiac and smooth muscles, you can't do that, right? If I want to slow down my heart, I'm scared. I'm like, oh, I shouldn't be scared anymore. I'm going to slow down my heart. You can't do that just based on willpower, right? You may be able to do it indirectly by, deep breathing and like thinking of nice thoughts, or you may be able to cause it to go faster by like thinking of scary thoughts, right? So you could do that. You can manipulate it that way, but it's indirect, right? Indirect, that control over your heart. And your heart otherwise is just pumping on its own. It's automatic. It's called this intrinsic system regulation. That is, cardiac muscle itself intrinsically just contracts by itself without any nervous system input. In fact, you take a heart out, put in an appropriate dish of solution, it'll just sit there and pump by itself. Our nervous system input is just to modify that rate. And then smooth muscle, similarly, is not controlled. If I want to say, hey, I need to speed up my digestion because I need to finish up this thing, I can't do that voluntarily, right? I can't constrict my blood vessels to direct blood flow. Like, let's say I have a major artery cut or something on my arm. I can't constrict all the blood flow to that area voluntarily, right? All that's involuntary, right? It's all done unconsciously, and it's not controllable by you in any kind of direct way, right? So that's the control over. And we'll get into that when we get into all these types of organs that have the smooth muscle as well as the cardiac. So that's control over it. We won't worry about the aerobics and anaerobics. So for cardiac muscle, if you were to look really close up to cardiac tissue muscle, right? So here's the heart of the wall, and these are the cardiac muscle cells. If you were to look really closely, they'd look kind of like skeletal muscle tissue with those striations, right? So you have these stripes are visible on cardiac muscle cells, which mean they have sarcomeres. They have that whole internal arrangement, right? If you look really close, this happens to be a cardiac muscle fiber, but it could be, for our purposes, it could almost be, with one exception, it could almost be a skeletal muscle. The reason why it wouldn't be is this center nucleus. Usually with muscle cells, the nucleuses are off to the side. But otherwise, right, it's got the sarcomere, it's got these T-tubules, the sarcoplasmic reticular, because in many ways it operates at this level the same way. But the difference is, remember, your your skeletal muscle fibers were arranged like, remember those endomysium and stuff, you wrapped each individual muscle fiber was like a piece of hair, and a fascicle was a bundle of hair. That is, they're all kind of bundled up next to each other, but they weren't directly connected. The cardiac muscle, they're attached end on end over here, right? So this is one, here's the one muscle fiber coming through, and then it may branch off, right, to attach to another cell. So they're going to be small fibers that are connected end on end to each other. And then we'll also see at these connections here, what are called the intercalated disks. There's going to be gap junctions, which will allow cytoplasmic stuff, and in particular sodium ions and potassium ions, because it's going to share everything so that they can have this electrical conduction across cells, these gap junctions. All right, so we'll talk more. I'm not going to get into anything like that for this test, but that's what's going on in this connection, right? They're strongly connected by those desmosomes and stuff like that, but otherwise they're connected end on end. They're going to form this big kind of sheet of muscle, right? So you're looking at, this is the heart tissue, right? Even though, you know, they may be running parallel with each other, they're also connected to each other in a couple different ways, right? So centrally located nuclei, usually one, maybe two, and unlike a skeletal muscle, which could have thousands of nuclei, involuntary control. They also, like skeletal muscle, if these cells die, you can't replace them as far as, you know, the conventional line of thinking goes. Once they die, you don't get replacement. So when you have cardiac infarction, right, when you have a heart attack and these have necrosis, you know, that's it, right? They don't replace themselves. Same thing with really damaged. So somebody's asking, the arrangement of myofilaments of actin and myosin give the striated look of skeletal muscle. Yes, that is true. Actually, we'll look at some histology. Yes, if you look really closely, then that's what you'll see. And I'll show you some other pictures of that. But in here, you can see too, the striations, this is the dark, this is the light and dark bands right here, right? And so these are all the myofibrils packed inside the whole cell. So, and they're all kind of arranged, stacked up in alignment with each other. And that's what gives that dark light, banding stripy pattern, this arrangement. And that's, you know, the overlap of the myosin and actin over here, the non-overlap, blah, blah, blah. What you can't really see is the H zone typically, you know, in any kind of micrograph. All right. So smooth muscle is not like that because they don't have sarcomeres inside. They do have actin myosin filaments that are kind of acting similar, but in a much more primitive way. So smooth muscles are kind of more like a primitive muscle style, right? They're not super complicated, but they do the same kind of thing in a way, right? There's no striations though, because there's no sarcomeres. They have like a small spindly shape. They're kind of like this. They might be connected end on end with gap junctions and stuff like that to connect them. But they're going to be arranged in a sheet, right? Like this, right? They're going to be wrapped around this. So what then they contract, they can manipulate the size of this lumen right here or make major movements, right? To squeeze, like say, food stuff through this tube, or over here, blood vessel, they can contract and constrict the blood flow, right? Create a higher pressure, or just maybe constrict the blood flow to a particular area. That's all involuntarily. And smooth muscle, unlike the other two, can regenerate if it's damaged, right? They do have some regeneration capacity. It's a smooth muscle. So this is just, this is a little more detail than you have to know, but just for your own interest, right? Here's that spindle shape, muscle fiber. Again, it's going to be connected to other ones through gap junctions. And when all these arrangement of actin and myosin filaments, as well as stuff within the other elements of the cytoskeleton skeleton contract, they're going to squeeze that whole thing. So when the whole thing squeezes a unit, you can get this squeezing force. And then again, red dot here, but this green stuff is the myosin. These are the actin. Instead of like Z lines, for instance, you have these dense bodies and they're attached to like parts of the other parts of the cytoskeleton, so that when they contract, they're going to make this whole squeezing force there. Those are really primitive, right? You'll see this in very primitive sort of organisms rather than our complicated sarcomere arrangement. Those are smooth muscle, right? So in general, right, these single, very long, cylindrical, multinucleate, obvious striations. Here you have also striations, not usually as pronounced in the pictures we have at least, but they're going to be kind of short, branching, joined end on end, whereas these are more of these like spindle shape, as they say, no striations, hence the smoothness. And last but not least, actually it is the least because you never have to know this stuff, but remember we had the arrangement, the connective tissue was creating this sort of tissue arrangement, endomysium around the cells, perimysium around the fascicles, epimysium creating this whole tube within a tube structure. You'll also have all that, but we don't talk about it when we get to these organs. But there is obviously always going to be connective tissue interspersed within all that. All right. So what I want to do real quick is go over the histology of all that. Right. So you were asking, you know, the striations, do the sarcomeres. Here's a close-up of a skeletal muscle. And, you know, maybe it's cardiac, but the nuclei, these small flattened nuclei here that are going to be kind of pushed off to the periphery is one clue. And if you span it out, then it's a little more obvious that it's not cardiac tissue. Right? But these striations, you can see they have a little bit of a, you know, they spell it out for you, A band, I band, A band, right? The A band is going to be separated by the H zone or H band. So from Z line to Z line is the individual sarcomere. But looking close up, all you could see basically is the dark and light bands, right? From this view right here. You can't see the H zone really. You can't see the Z lines until you get zoom really far in. But that's your skeletal muscle from a longitudinal view. The other one would be the cross section. This over here is cardiac muscle tissue. You see these big, kind of fatter, more oval-shaped nuclei rather than like the squashed out one. And if you look really closely, this one isn't super definite, but there are striations in there. Right? But this is one piece, and you can see how they branch off and connect to the next muscle cell. And you can even almost make out the intercalated disks on these right here. But you can see all this better. Right, so that's your cardiac muscle. And then for your smooth muscle, right, this is what we're going to kind of encounter quite often. When we get to, say, your digestive system, this is a piece of your intestinal tract here, your small intestine. And you're going to have those villi. Here's going to be all the food, and surrounding the whole tube, you're going to have this outside layer of smooth muscle that are going to move stuff along that tract, right? Or squish it up and mechanically break it down. So in this case, you usually have a couple different layers. You have one that's running lengthwise, one that's running in a circular mode. This is actually the circular one. This is the longitudinal, but this is a cross section. Hard to tell, but these longitudinal ones, right, these are smooth muscle running along here. So those are smooth muscle. Another picture of your smooth muscle. This is your gallbladder. We're never going to talk about the gallbladder. Is that true? No, we do talk about the gallbladder, but we don't look at it histologically. But I'm using this because it does have nice samples of smooth muscle. These are ones you could actually see the outside shape. Right? You can make out that whole spindle shape on the individual muscles, right? So there's the nuclei. These elongated things right here. So this is a nice example of the smooth muscle, which is why I use it. These are longitudinal, and those are crossing. I usually show for this, I'm going to be showing the focus in on the longitudinal. All right, so that's it for my mini lecture on these different types of muscle, right? You kind of just want to compare them, take a quick look, understand these bullet points over here as the differences between them, right? Location, and then a couple of the characteristics of them.